Good afternoon. Welcome to North American Neuromodulation Society April Journal Club. My name is İknur Telkes and I will be moderating today's session. All right. Well, as we begin, I want to send my thanks to NANS Engineering and Science Program Committees for their continued support, and my colleague, Dr. Irina Da, for her dedication and commitment. And of course, many thanks to our coordinators for facilitating today's event. Well, for those who are joining us for the first time, I want to share our objectives briefly. We've been organizing this initiative since 2020 to bring together neuromodulation experts and experts from the neuroscience, engineering, and clinical fields with interest in neuromodulation. Provide a platform for trainees to present their newly published scientific papers, showcase the technical advances of each research group from diverse disciplines, and discuss the various aspects of neuromodulation research. And we hope these meetings will become an excellent platform for new exciting collaborations among scientists, engineers, and clinicians. We have several ground rules. This is a 1-hour webinar. We will have 40-45 minutes presentation and 15-minute discussion at the end. Attendees will be in listening mode throughout the presentation, but you can type your questions, comments to the chat box anytime during the talk, but we will address them at the end. Experts' experiences or comments on the topic are all welcome. Please weigh in if you want to. And finally, the webinar will be recorded. You will be able to access it through the NANS website. Well, I don't have any relevant disclosures to today's talk. And well, the paper we will be discussing today is entitled Local and Distant Cortical Responses to Single Pulse Intracranial Stimulation in the Human Brain are differentially modulated by specific stimulation parameters. With this paper, through this paper, we will define the limitations and challenges in understanding how direct electrical stimulation changes brain activity, identify how stimulation parameters matter, including the type of stimulation done as well as the location. And finally, we will define how the fine-scale location of the direct electrical stimulation can shape brain activity. Now, it's my distinct pleasure to introduce today's speaker, Dr. Angelique Paul. Dr. Paul currently works as an instructor at the Department of Neurology, Massachusetts General Hospital in Boston. She is carrying out research in neurophysiology, neuroscience, and behavior, focused on understanding the neural underpinnings of behavior, leading to treatments in neurological and neuropsychiatric disease. Through undergraduate and master thesis work at Cornell University, PhD at the University of Arizona, at first postdoctoral position at the Queensland Brain Institute in Australia, and work at MGH, Dr. Polk's approach has been built on multidisciplinary approach founded on technological development and utilization in the context of robust science-predicated and careful observation, followed by hypothesis testing. Dr. Polk currently works with intracranial data to study neural and cognitive processing in the human brain, recorded using intracranial electrodes, including investigating the mechanisms of how direct electrical stimulation alters neural processing and behavior. So without further ado, Dr. Paul, please take it away.
Thank you very much, İknur. I really appreciate being able to discuss this paper with this group, and I really appreciate the invitation. It's a fantastic neuromodulation journal club, and I'm excited to be able to sort of talk about all these kinds of, cool aspects of our work with this crowd for sure. So as İknur sort of referred to, I'll be discussing this particular paper that we just published recently. We were very also lucky to be able to get the cover on brain stimulation for this work. But first and foremost, I should thank the team. I work in the Cash Lab with Sydney Cash and with the lab manager Yang Chu, and we get to do a pretty amazing amount of work working with humans and intracranial recordings. And this also, was not possible without the, massive amount of work from some incredible fellow researchers, Rena Zellman and Brittany Crocker, and Ishita Basu and Jackie Peralta, but also with the help of clinicians and neurosurgeons and neurologists, as well as psychiatrists, to be able to ask these kinds of questions about how stimulation modulates human brain activity. And I will have to say this data has been gathered over years. I think the first data set's 6 years old, really. So this has been a lot of hard work to be able to sort of bring, these questions together to be able to address an important question regarding direct electrical stimulation. Direct electrical stimulation, as this this particular audience would no doubt know about, is used in a wide variety of neurological and psychiatric diseases, whether it's therapeutically or to be able to sort of map neural activity, such as in the operating room. And it's not always effective though, and it can be highly inconsistent. So we actually posit that electrical stimulation could be used for much more if we really understood how it works, particularly the input, the stimulation input, as well as the output brain response relationship and the time scale of this particular effect. And of course, the interrelated challenges to be able to sort of dissect this information underlie this inconsistency, because, mainly because treatments are often trial and error, as effects are not always understood on a mechanistic level. We don't always know what cell types and cells and populations are actually doing with stimulation, in part because the stimulation parameter space itself is almost infinite, and I'll get into that a little bit. And there's also variability in the underlying networks in between individuals. So just to give you a sense of what we do in the CASH lab, we actually worked with direct electrical stimulation in 3 different main domains. One of them is in the acute domain, where we actually record neuronal single unit and local circuit, local field potential activity during and after stimulation in the operating room, often using microelectrodes. The other space that we work on is the epilepsy monitoring unit, working with patients with intractable epilepsy who are implanted with leads for about 10 days to be able to identify seizure onset locations in the semi-chronic setting. Another sort of setting that we work in is we're looking at activity during and after stimulation with a responsive neurostimulation device for chronic implantation and a closed loop treatment of epilepsy. And except for this last data set, most of the data we look at involves very short bursts of stimulation, 233 microseconds to 2 seconds, with ongoing sort of neural recordings to be able to examine stimulation effects. And really, an important piece of this is also that we're, today I'm actually just going to be focused on this semi-chronic setting, because that actually allows us to look at sort of like, stimulation over a short period of time, but not necessarily, in a longer term chronic approach. And what we've been doing in the past 5-6 years is really trying to understand this central question of input and output, and examining directed electrical stimulation, within sort of 3 main functions: understanding what stimulation is doing in the human brain and how it works, in terms of certainly in the mechanistic aspect; use it to probe networks in the brain and various different sort of aspects, as well as, ask questions of whether or not it could be developed for therapeutic uses. This is, these different images are some of the different papers that we have been publishing, especially in the past 2 years even, to be able to sort of indicate that we're actively trying to examine what is stimulation doing, how it works, and whether or not we can make it better. Because, a big part of this is, can we use this to better circumscribe the input-output relationship in these systemic, types of studies. And relevant to that, so I'm actually just going to be working on, discussing this one particular paper, the local and distant cortical responses to single pulse electrical stimulation. And really sort of how we can look at how the input, the stimulation parameters, can change the output. I'm also going to play this little movie, mostly because it summarizes the entire, my entire paper, this entire paper, but it also gives you a sense of the kinds of neural responses that we would actually observe. So we generally stimulate at bipolar pairs of electrodes at these 2 neighboring pairs. What you're going to see is the stimulation waveform. The input is sort of changing shape over on the left, and the stimulation output, the response over here in blue, is actually changing its shape with these different kinds of shifts in the sizes, the shape, the amplitude, the duration, etc., as well as the location. So a big part of this is also sort of dissecting not just that we can change the stimulation input parameters, but we can also change the location as its own parameter to ask questions about how we see different kinds of responses along the a implanted lead. So really, this video, summarizes the big main take-home points of like what we were trying to examine, and the rest of my talk will actually be examining, okay, what exactly is this input-output relationship? Now we tried to characterize that input-output relationship in in terms of the quantification of the responses. So as you just saw, we're looking at these sort of generalized waveforms that happen after single pulse stimulation. We're going to focus on the single pulse stimulation because it's been used for sort of mapping networks in the brain in a number of different sort of domains. And I just wanted to show this particular example, which was actually a really handy example in a specific patient where we stimulated at different locations relative to the gray and white matter. So the gray matter is indicated here by this gray boundary, so the gray-white boundary. And then the cortical outer portion of the cortex is indicated by the black sort of dots here. And at the stimulation location here, relative to the other channels that are recorded in the brain, we actually see sort of an ongoing spread in response whenever we stimulate it at this particular point. Now, if you just shift the electrode to a different location, we found a more localized sort of like stimulation response. And yet another different location, almost no sort of spread of response. But what you'll actually notice here is that not just that location matters, but actually there are consistent waveforms that we would find with this type of single pulse stimulation called the cortico-cortico evoked potential. And this particular potential has a particularly distinct waveform, has a long history in the literature for being used for understanding sort of neural networks, including seizure networks or epileptogenic networks. And the idea here is that you can actually, subdivide portions of this measurement into, at least 4 different measurements. In this case, what we chose to do is look at the peak amplitude, overall peak amplitude of the response, area under the curve, overall kind of like summation of the voltage response. Looked at N1 peak, which is that early sort of peak response, and then N2 peak, which is that later peak response. And we'll use these 4 measures to then characterize what is, sort of the input, stimulation parameter input, relative to the neural response output that we can see in these kinds of dynamics. And what we then did was we were very fortunate and very lucky to work with some amazing participants and patient participants who consented to this work in the epilepsy monitoring unit, which is specifically 52 individuals. And we asked, okay, if we stimulate at different locations, which is illustrated in the dots in the upper figure, what exactly is going on with the different kinds of responses? And one thing that we couldn't, once we, started to gather this data, there was something striking that we couldn't necessarily parse out without actually, having a full, accounting of the entire data set, that there was something different about some, local responses versus distant responses. And the lower figure is to illustrate that. If we then sort of indicate sort of the recording electrode, which we had, in this data set, a total of 719 independent stimulation sites across all the participants with over 6,500 recording sites, and we took a look at the recording site distance, Euclidean distance relative to the stimulation site. We found that there was this drop-off, this very clear drop-off that would actually happen, really within an inflection point around 15 millimeters away from stimulation site. So if you actually are recording further away, there would seem to be a very much sort of flattening of the response. Whereas like if you're right close, especially at 15 millimeters, there was something that was a striking response profile right there. And that particular distinction made us sort of decide to actually have sort of a separation between a local response and a distant response. And there's a number of different reasons to that. And in fact, a paper by Crocker et al., a previous paper we had, actually sort of identify that there is a reason, there are some interesting reasons regarding gray and white matter tracks, that actually there's a reason to actually have this kind of distinction. But in this case, we decided to have that cut off mostly because we had a feeling there was something different between these 2 different scales. So the outline of the paper, or at least what we actually tried to hypothesize regarding the stimulation input and these different parameters and the ultimate sort of cortico-cortical evoked potential output, was that we had first hypothesized that increasing the duration of the pulse, the single pulse, would actually engage more of the distant brain network or produce more of a response. That's not surprising. You inject more current, you get more of a response. We also sort of hypothesized that you can have increasing amplitude of the stimulation and have a linear increase in the voltage response. This actually comes from a paper that we published where we found that there was a linear relationship between the neural response to trains of stimulation and injected current. That's what we hypothesized here for the single pulse as opposed to a train. We also hypothesized that there was as a base understanding or base sort of like hypothesis that there would be a linear relationship in terms of when you move the stimulation site closer to white matter, we'd get an increased response and moving away from gray matter, as well as because you could be engaging more of the axonal network, as well as sort of a linear relationship relative to orientation relative to the cortical column. And finally, of course, and something that does come from a few of our papers previously, that there was something different for the different brain regions stimulated. So I'll go through and step through the evidence that we have for this and whether or not these hypotheses really pan out for the rest of the talk, because this is how we actually did approach this data. I approached this data in such that we did have these original hypotheses, and we wanted to ask, could we even answer it with these kinds of data sets? But so in other words, we're starting with the duration question and the original hypothesis there. Yes, when you increase the duration of the stimulation, you actually get bigger responses, and they do seem to sort of spread further. So it's going to orient you to this particular slide, and you're going to see this pretty consistent for other data. The rest of the slides is that we have the local response, basically the local recordings, less than 15 millimeters away from the stimulating electrode. And then the distant recordings, which are further than 15 millimeters from the stimulating electrode, which I indicate in gray and black here, inverse relationship. So that basically the idea is that you still essentially have a significant response that's higher whenever you increase the duration of the input pulse. So that happened for this particular data set, and so that particular hypothesis made sense, and there was an increased pulse duration response in general. When we then took a look at another stimulation parameter, which was increasing amplitude. In this case, it was increasing amplitude by 0.5 milliamp steps. So we actually had and I'll actually mention for both the duration and the amplitude measures, we had a smaller data set out of the entire data set of N of 52, only because of time constraints, because the duration and the amplitude measures tended to take longer. So therefore, we only did that with a subset of the data set that we used for the entire paper. But we were still able to ask across participants and ask questions about what exactly happens when you increase the amplitude of the current injected. And you see, generally an increased response. That makes a lot of sense in these particular figures for each patient, which is, has a patient designation indicated by HPA32, et cetera, and for different sites, which is indicated here. You see that the black line is the average of the response across 10 trials, and the gray lines are the individual trials. You overall see a very consistent increase in the response with increasing amplitude injected current. But the original hypothesis was that it was a linear relationship. Well, we didn't find that, which was interesting in itself, because what we found from the trained data is that increasing amplitude had a linear relationship with increasing voltage response. Here, for the single pulse data, we found a nonlinear relationship, really more of an S-shaped curve. So this is that peak response. It would actually sort of like steadily sort of rise and have a fast rise sort of period somewhere around 2 to 4 milliamps, and then settle down around past like 5 or 6 milliamps overall. And that meant that our original hypothesis didn't match, but also we wanted to be able to ensure that we could test this using specific quantifications. And so what we did was we used generalized linear models, which allowed us to then fit the data such that we could actually test whether or not it was a first order generalized linear model fit, which was polynomial fit, which would be a line, a straight line, or if it would actually be better fit by a second or third or fourth order polynomial fit. And to then sort of identify the optimal sort of fit for these kinds of data, which are just represented by the scatter plot over here, we then used IKK information criterion to identify the minimized fit for each individual line. And by and large, for the amplitude measure, for sure, it was actually a second order polynomial fit best described the data. We also did a number of other measures, including linear correlations, et cetera, and they didn't actually fit as well as basically this polynomial fit. And so more or less, it's a nonlinear relationship. It may not surprise many of you, but it did actually surprise us, considering our past data sets that we've actually examined. So as and so basically, now taking this particular model, we took a look at the local, so basically the black line sort of responses for the different measures that I had mentioned earlier we used for the neural response. We found it was clearly just a nonlinear sort of relationship and rise in the response. And also when we looked at the distant responses beyond 15 millimeters away from the stimulation site, we also found this nonlinear relationship. So this, again, sort of emphasizes that we don't necessarily have a linear relationship going on between current and the overall responses. I wanted to actually take a moment here where I actually brought together, in this previous slide, I indicated that we were looking at just local versus distant as sort of a either or. If we then look at, a series of distances away from this site of stimulation, not just subdividing distance, from the stimulation into one or the other, we actually find that a lot of the effects in terms of increased duration, as well as increasing current, were also generally sort of local in that they were generally, less than 30 millimeters around the stimulation site. So basically, as you increase current, the significant effects were really kind of happening less than 30 millimeters away, and then further away, it didn't really have as much of a significant effect. When you're increasing duration, the same sort of interesting sort of result, which meant that like you can hammer, you can actually increase like a lot of these things up to a point, but you may not be reaching those distant sites the way that you actually sort of anticipated or would have predicted if it was just a sort of a spread of current through, a saline volume, for example. So getting back to this idea is that basically we were kind of wrong on both hypotheses so far. I'm accepting of that. That's what we're doing, where this is the science. This is what we try to address. But this nonlinearity, I think, will be important for understanding sort of especially cortical stimulation and its treatment in terms of modeling and understanding what's going on with these dynamics. But as I also sort of hinted at, is that we're not necessarily putting a stimulator into like a water balloon. We are working with a network. We're working with gray and white matter. We're working with a lot of other sort of aspects that are complex and amazing, which is represented by the human brain. And that includes location relative to gray and white matter. So we have quite a bit of interesting evidence that actually kept on showing up that we wanted to ask a question of. And regarding if you stimulate it in the white matter versus the gray matter in the cortex, specifically, what exactly happens in terms of the neural responses? And I have to say, there's been some fantastic papers that have been coming out in the past, for cortical stimulation in the past, about 2-3 years from various other groups, essentially, sort of identifying that, where you are relative to white matter makes a huge difference. And this is just, again, essentially just replicating or reiterating or emphasizing this important relationship. And with regard to that, I wanted to, take this hypothesis that location relative to the gray-white boundary, especially if you cross a gray-white boundary into white matter, you can get more of a response. The original idea here was to be able to sort of use two different types of measures independently to be able to sort of test this original hypothesis. So I used one of them called the categorical or volumetric approach. This was just a way of essentially, you take electrode locations, you identify where they are relative to the white matter volume. This is all geometric approaches in general. Or relative to the, if they're in the cortex, which is indicated by the blue, or if they're sort of in the outer cortex, which is in a sulcus, or sort of like further out, relative to the peel surface. So that was the one approach to be able to then identify that we have recording electrode or stimulating electrodes that are in the white matter, in the cortex, or elsewhere. The other metric that I used, which was more of a, what I call a more continuous or distance approach, was actually using Euclidean measures to measure the distance of the nearest gray-white boundary, basically the boundary between the gray and white matter, and ask, okay, can we do a continuous measure of sort of, wherever things were in the negative values, that'd be closer or within the gray matter, and then whenever things were in the positive values, that was be sort of represented along an axis in white matter. And this was just a way or convention that allowed us to sort of like do a, take all these types of electrodes, and it can be tons of electrodes, to be able to sort of have a consistent measure of distance relative to the gray-white boundary specifically. And so we could then do these two different measures independently using the same data and ask, okay, do we see, and how can we test this idea that there's a white matter, sort of responsive or stimulation responses that vary between the two different kinds of measure, between gray or white matter. And so regarding this more categorical approach, and again, sort of just down here reminding you, these are the different kinds of measures that we use to sort of characterize the overall responses. And I just want to remind you also that this is all based on stimulation location, not necessarily the recording location. That's actually a whole other paper. I'm not going to touch it right at the moment. But with regard to stimulation location, what was interesting was that whenever we were looking at the white matter stimulation specifically, there was a significantly higher sort of response at distant sites, basically sites that are further away from the stimulating electrode than compared to cortical stimulation. But if you look at local responses, it wasn't as striking a difference. And that was very interesting, that basically a local response doesn't always differentiate between cortex and white matter, but distant responses do. So that in itself was interesting, because we also again took a look at not just this categorical, sort of boundary at 15 millimeters, but also this, more continuous measure of distance to the stimulation site. And this difference between white matter and cortex was not necessarily, again, not right next to the electrode, but really in this sort of middle range of about 30 millimeters away, that there was actually a real distinction between gray and white matter stimulation. So this is a very interesting keeping in terms of like whether or not this could be used for modeling and understanding what's going on with the white matter versus cortical stimulation, and whether or not you can even say target particular types of stimulation at certain distances using this kind of information. So the other thing, the second measure, which was done independently, which was actually to sort of do this more continuous measure of distance relative to the gray-white boundary. And as you kind of hinted at from the previous slide, that actually you get closer into the white matter, you get an increased response. In this case, is the proportion of responses channels. And here, what was interesting for us was, and this actually was again reflected also what we saw from the volumetric side, is that there was something different happening locally than there was at distant recording sites. So here, what I'm emphasizing here in black are actually the local sort of responses here. And you see basically it kind of actually curves and peaks at the gray-white boundary itself, not necessarily in the white matter or the gray or the cortical gray matter. That in itself was very interesting, even across all the different measures that we were using. Whereas if we then sort of essentially zoom in this gray line down here, we take a look at the stimulation at distant sites, we see a very clear, nice nonlinear increase, almost exponential increase of responses when you are stimulating in the white matter versus the gray matter. So there is actually a flip, also like a sort of happening inverse of what is happening in terms of local effects versus distant effects. And this also was a little bit reflected when we then looked at the distance relative to the stimulating electrode, instead of just the dichotomy kind of approach. And then of course, as I hinted as previously, we also looked at orientation because that location and orientation have pretty important sort of interactive roles, really, in terms of like stimulation and relative to the cortical axis, and or in the case of like hippocampus, for example, the hippocampal axes. And what we found here was a mostly purely local effect, in that basically when the stimulating electrodes, the bipolar pair of stimulating electrodes was perpendicular to the axis of the cortex, you'd have the strongest response. It's a nonlinear relationship, but had no real effect on the distant effect responses. Couldn't find a relationship there. So that again sort of emphasizes that we have a very different kind of response world in terms of local versus distant responses overall. And so this just emphasizes that again, when I originally sort of hypothesized that it was linear, I wasn't really certain about that hypothesis, but we did actually test that it was definitely not, and show that it definitely was not linear. But we found that like there was these interesting relationships and interactions between sort of stimulation location and orientation, as well as the gray-white boundary and the cortex. And finally, what I'm going to end on is sort of a central question of like, is this the same across the different brain regions? And that's an important piece of this puzzle that, especially for cortical areas, which can have wildly different connectivities across the different brain regions. What exactly is going on with these stimulation parameters? In this case, I'm going to focus on stimulation parameter of location relative to gray-white matter, because we were very lucky and very fortunate to be able to sort of work with some amazing patient participants and then also be able to sample a decent amount of space over these years of gathering the data. While we didn't actually sample a lot of the sort of parietal and occipital lobe areas, I'm not going to actually focus on them, mostly because I just didn't feel comfortable sort of like including them in the kinds of analyses we're here. So focusing on the lateral frontal lobes, the cingulate cortex, and the temporal lobes, we found some interesting sort of interactions between location and the brain regions themselves. So what we found was that we would again do this polynomial fit and ask questions of like, is there a significant relationship between the gray-white stimulation location relative to gray-white matter boundary and the brain region? We found that the lateral frontal lobe actually had a linear relationship, which apparently we're partially right on that original hypothesis, that basically there was an increase in response with that was actually a linear relationship, as you go further into the white matter, particularly in local responses in the area under the curve. It was a little bit more fuzzy with some of the other measures, but it was a very interesting distinction compared to the temporal lobe, which is on the right side, where basically the temporal lobe exhibited this peak at the gray-white boundary for local responses, but not necessarily for, but not this linear relationship as the lateral frontal lobe. What was also interesting is that, we had a fair amount of data for the stimulation of the cingulate cortex. And while I fit curves here to sort of illustrate there might be something there, the curves are not significant. They're actually not, sort of stably a significant relationship between the location relative to gray-white matter boundary and cingulate cortex responses here. So this is very interesting in that basically one brain region doesn't follow any of these sort of like predictive rules, whereas the other two seem to have two different types of rules for these kinds of overall responses. And that in itself, sort of speaks to, are we actually, each time we look at sort of overall brain regions, should we actually start to really kind of make certain we at least dissect which part of the brain we are actually sort of studying this in. And to also illustrate this, we are, in addition to laying this out in 3D, such that we have a look at the distance relative to the stimulation electrode and the gray-white matter, we also see that there are, again, this landscape is different between the different brain regions. And so really, we tested a bunch of hypotheses. The only hypothesis that really sort of panned out was the last one, saying that different brain regions are different, which should not surprise everyone. But at least something came through for that. But I'm actually going to quickly kind of scroll through the rest of these, mostly because I have a little diagrams to kind of bring home the take-home message. Was that we really were able to find that increasing stimulation duration for single pulse electrical stimulation did actually sort of increase the responses sort of in this middle distance, local and middle distance from stimulation. Not necessarily as far as I hint here, but it's some interesting response there. Difference that the relationship between injected amplitude or current amplitude and responses were nonlinear, not linear, like we actually sort of hypothesized originally. That there was an important and surprising relationship between the location relative to the gray-white boundary and local and distant responses. And these responses were generally nonlinear, whether you use the volumetric or the more continuous measure. And there was a nonlinear relationship between orientation and local neural responses as well. But these needs to be sort of taken into consideration that there are maybe ideal sort of locations for be able to induce stimulation responses in the different brain regions that can vary depending on which brain region we're actually stimulating. And that's an important piece that we were surprised at, especially for the cingulate, but also for the other two brain regions that we focused on. And so the future directions for all this work is really sort of being able to move on to not just we talked about sort of this geometric relationships of electrode locations and relative to the brain structure. But one thing that we didn't really talk about, but we are studying actively, is the underlying network structures, such as the functional, affective, and structural connectivity underlying the brain, such that we can look at sort of like white matter tract mapping, et cetera, and how that actually maps to what these particular response profiles are into the terms of the input and the output of the response. Especially because right now we're finding out that single pulses are doing something very different to what trains are doing. And that's an important piece of this, since trains of stimulation, which are represented by this, you know, this sort of like burst of stimulation up here, are really what are being used therapeutically right now. And we're also actively investigating the cellular basis of stimulation responses, what underlies these stimulation responses, using high-density extracellular recordings in vivo and working with Mark Harnett at MIT to look at single-cell patch clamp responses and understanding what exactly individual cell types do, such as excitatory inhibitor neurons, in terms of the overall responses. And finally, a big part of this push and a big part of our active role is trying to ensure that we get the data out there to be able to answer, you know, or ask different hypotheses that we would not have actually thought about. So we are actually actively trying to follow and engage in open science data initiatives and be able to share it. And so this is a right here website of where we've actually uploaded the data from this particular paper. And feel free to reach out to me if you have any questions about it. And then the significance of the study, you know, with regard to specifically neuroscience and medicine, is that we are really, you know, we do believe that, you know, direct electrical stimulation could even be used for more than it is being used for right now. But it would help if we understood the input and output stimulation rules and to allow us to provide sort of informed stimulation paradigms, particularly in terms of the understanding of the mechanisms underlying them, to be able to shift the brain into different network or cognitive states, such as in the, you know, treatment of epilepsy or neuropsychiatric disorders. And, you know, we believe that understanding how stimulation operates on the level of single cells to the whole network could lead to the use of stimulation in unique ways, possibly, you know, inducing plasticity or the treatment of such disorders as TBI and stroke. And we're hoping these kinds of studies and these kinds of investigations would allow for the informed design and models of neuromodulatory therapeutic tools for a wide variety of neurological diseases. Maybe, you know, I don't know if it'll be a shift, but so much as, you know, allow us to be able to start to predictably interface with the human brain, such as in BCI. And I wanted to thank, you know, an amazing team, an amazing group of people that have made this possible. It's, you know, really there's a lot more people that should be on here, but a big, you know, sort of major contributors to our entire investigation is actually the patients who participate in the studies. You know, they are incredible and an honor to work with. So thank you very much.
Well, thank you, Dr. Polk. It was a great talk and very stimulating. And thank you for walking us through it. And I guess we can take questions. Do we have any? Let me check. All right. While waiting for questions from participants, I can move forward with my questions. So I guess my first question is related to electrodes. So do you think, I assume you use the same type of electrode in all patients, right? So do you think if, or maybe not, I guess my question is, if you use electrode with different contact spacing or a smaller contact size, or, you know, depending on those parameters, do you think your findings would be different, especially your 15mm threshold would be different if you use, or maybe if you use tripolar stimulation, which is inducing more compact stimulation, you know, sim area. So do you have any comments on that?
I think there is. I think that there's a, I completely agree. I definitely think that there will be an effect up until a point. I have like part of one of my slides is actually these attractor states kind of things. I think that basically there is a range of contact sizes that, you know, this range you'll get this kind of response. This other range you'll get a different kind of response. Tripolar I've never messed with, so I can't speak to that. But for the bipolar stuff, there seem to be certain ranges that we got very similar responses. In fact, such similar responses that were even like predictable across non-human primates and humans. So like that, even with like, there's basically certain ranges that I think that if we were within that buffer, we'd be very similar sort of response profiles. And then if we skip to another sort of range of really smaller electrodes, it'd be a different kind of like response profile. I think that would definitely be the case for the local responses. I don't think this is me waving hands literally right here. I don't think it'll be as much of an effect on the distant responses. I think the polysynaptic aspect of the distant responses may not matter as long as you inject enough current at the site here. The far off doesn't matter what it's going to do with that size of the electrode. And that actually is the reason I say that is I think that's because based on the orientation question, it didn't matter what orientation you're there when you were recording over here. It was actually, I think that that's a case where the contacts and context or specifics may not be as sort of effective in being able to produce those distant responses. I think in those cases, it's actually mapping to the white matter tracks or using DTI, for example, or the very nice sort of like tracula kind of approaches to say, okay, actually, if we want a distant response, we just need to move the electrode a little bit over, and we're catching that highway, and it won't matter what the electrode size is, as much like in general.
Okay, thank you. We have a question from David McMullen. How do you relate your results, boundary locations having largest impact, for example, with traditional DBS targets?
It's a tough one because, I mean, it's a great question. One of the biggest issues is the fact that I'm working, the majority of the data we worked with was cortical stimulation, and the traditional DBS targets are subcortical with the exception of some of the depression stuff. And there's some pretty cool studies, from Mayberg, et cetera, showing that if you're actually closer to the white matter, you may be able to sort of induce some of these distant effects. But it's a case where I'd love to sit down with some some people working in DBS to really kind of see how different is this when we're talking about cortex, like lateral frontal lobe versus obviously subthalamic nucleus. I have a feeling it's going to be a lot of the rules, maybe even more nasty in terms of non-linearities with cortical stem, but maybe not. We need to bring the data together, do open science, have open data source, and really kind of dig down into that.
Thank you. Another question, I guess my question is about the patient cohort. So how uniform was your patient cohort, like in terms of location or type of epilepsy or epileptic zone? Maybe you can comment on that for the people who are not working on epilepsy field.
There is a lot of variability. Yeah, with the epilepsy, there's a lot of variability, and there's also the case where a subset of the patients actually had resected tissue, so they were actually missing parts of the temporal lobe, or there were lesions. We actively, for our stimulation, we actively tried to stimulate in areas that was far away from the epileptogenic zone based on what the clinical team identified. So that was our high amount of consistency. Regarding location, I have to say that I didn't think there would be consistency, but we found, especially with a trained stimulation, but also a little bit with the single pulse stimulation, that neurosurgeons have patterns. And so we actually would get, if I sort of looked at the MRI relative in the electrode location in the native space, it would be very similar across some patients. We would actually see a lot of consistency across the board. Again, far away from the epileptogenic zone. I can say that there's a lot of variability, but also the fact that we have found that we were able to predict neural responses from using one data set, predicted in another data set in a different patient. So there is actually a remarkable amount of conservation of the responses overall.
And
but
I guess, yeah,
it's a lot of variability of like what exactly is the type of epilepsy, et cetera. It's extremely variable on this.
Maybe a follow-up question to that. You touch base with this in paper, but all patients took medication before the recordings, right? So do you think responses would be different if they were, they wouldn't take medication? Because at the group level, the responses are pretty robust and consistent across patients, even though they show different demographics or diagnosis. So how about if they were under no medication? Do you think it would be different? Or can you comment on that?
We don't gather data for like without without them being on some medication. So like that's in our IRB protocol currently. So I can't speak to that on data that I haven't gathered. My hypothesis is that areas that are more irritative, like basically that are more active with epileptic or genetic activity, I think they would be just more active and more inducible into with stimulation. As to sort of, there's actually some amazing papers though out there that I've never touched, but actually using single pulse stimulation to identify and map epileptogenic zones with the patients on and off medication.
Mm-hmm.
And there is a difference in responses, but that has to do with also like sort of the zones as well as the medication.
Mm-hmm.
I think it might be different. I don't know though, unfortunately.
Okay. I mean, I'm coming from DBS and, did some Parkinson's disease research, and medication and stimulation affect, the regions and the patterns differently. So that's why I guess I was wondering if it would be the case for epilepsy.
I think it is, but I think it also depends on the medication. And there's so many different types of medication to be able to sort of squash sort of like seizure symptoms that I have a feeling it might even be more complicated than that. So like, unfortunately, I can't speak to it. We don't work in that space yet.
Well, we have a question from Nicole Provenza, from our previous speaker, former speaker. All right, let me read the question. It's a long one. In the white matter just superior to ventral striatum, which is the target DBS region for OCD, we see extremely variable behavioral responses when stimulating at different contacts that are 1.5 millimeter apart. For example, at one contact, we might see smiling and laughing, and at an adjacent contact, there is no behavioral response at all. Do you think it is possible that the local effects of stimulation act over a much shorter distance in these deeper structures?
I think it's a good point. I think it goes back to me talking to a bunch of DBS people about it, though. I think I agree with you, Nicole. Via this design. I think that there's some very fine-tuned and amazing DBS. It's not just actually in OCD sort of literature. I mean, there's some really good papers out there of looking at movement disorders where just even a half a millimeter makes a difference in being able to sort of in treatment, or even less. And it has to do with this white matter tract mapping. So that is a future direction that we want to go in as to how local versus distant things are. I think there's a lot of really cool questions of taking this sort of very simple geometric approach I took, which was this paper, and then merging it with the white matter tract mapping. That I think them together may actually help us answer some questions that are just when you're looking at just one side of the coin versus the other, you're not answering the full question. I think so. That's not really an answer so much as I think it's a great point, and I think it's an important point, and we should have more recording electrodes when we are stimulating, honestly. So.
We have another question, this anonymous question. How did you justify for IRB the need for stimulation in these patients? How did they let you do that cortical stimulation?
So RNS is actually involving cortical stimulation in several different cases. So it is actually a part of a therapeutic sort of treatment for, not all epilepsy, but a certain amount of epilepsy. And that is one of the treatments for, within RNS. Single pulse stimulation as well as train stimulation are actually both used for clinical mapping. That is actually a part of their protocol. In fact, single pulse stimulation in several hospitals in Europe is actually used as a part of their clinical workup to identify network mapping of these individual recordings. So it is in fact a part of the clinical domain. What we are doing is actually, this is what an important piece of justifying to the IRB was that these particular patients and participants, we actually use stimulation to be able to sort of understanding the network, but we use such low levels of current relative to clinical mapping that it is we calculated out. It was actually several log steps below the amount of current that's actually used for clinical mapping. We are using such low levels that in fact, what Nicole referred to earlier, we don't even get behavioral effects. The patients can't feel them either. So this is a investigation to understand the brain, understand the stimulation mechanism, but also this is well within the bounds of what is even used in the epilepsy monitoring unit for clinical treatment, or understanding where exactly areas to resect and what areas are functional, such as language and motor areas. What exactly is going on within the network and the epileptic network in the brain.
Thank you for filling out. I don't see any other questions at the moment. Maybe we can give another minute or so to our participants. If you have any questions, you can type it to the chat box and we can address it. I guess my last question would be about the, N1 and N2 characteristics. You mentioned that it is pretty commonly studied in the literature. Can you just elaborate on their functional use in your research or among your findings?
Yeah, N1 and N2 is a rough one, mostly because right now it was characterized, about. Yeah, it's a distinctive response profile. You see it across a lot of different individuals and a lot of different patients and in various different settings. The biggest issue with N1 and N2 is it's not as robust as people have communicated. So there's actually sort of not just myself, but other groups were finding that that might not be the best way to characterize the neural response. The original idea was that N1 is possibly the excitatory neurons responding, and N2 is the inhibitory neurons responding, or the rest of the network responding back. In terms of sort of the basically stimulation responses go out, and then the response from the rest of the network is actually N2. There's a lot of different ideas of what it is. It's not as consistent across brain regions as people have communicated in some papers. So it's a case where we actually have an entire study going on the side of saying, okay, there might be other better measures of this to be able to sort of dissect this, and especially relative to single neurons, or what individual neurons are doing in this network, because that's we really want to get down to the mechanism of what's driving this. And N1 and N2, that particular waveform shape is very hard to find in the temporal lobe, but you find a lot of it in the lateral frontal lobe, for instance. So I think it's a difficult measure. That the reason why in my paper we also went with those two other measures, which was peak and area under the curve.
Okay, awesome. Well, thanks so much. We don't have any other questions, and I guess we can close the session. Thanks so much.
Thank you very much. It was a pleasure.
All right. Take care. Bye.
Bye.